Hello, I'm Daniel Kuritzkes from the Division of Infectious Diseases at Brigham and Women's Hospital and Harvard Medical School. And today I'm going to be giving you a brief talk on HIV cure through the ages, focusing on the importance of cure research in very young infants. These are my disclosures. As is well known to this audience, the timing of mother-to-child transmission of HIV depends very much on a number of factors. And there are really three different times when transmission may occur. In utero, by the transplacental route; peripartum, believed to be by the oral or mucosal route; and then postpartum through breastfeeding. There are specific diagnostic criteria for establishing the timing of infection. Infants or babies infected in utero will be HIV DNA and RNA positive at birth. Those who are infected peripartum will be negative at birth, but become positive within a month of life. And those who are infected postpartum are negative at birth and at 1 month, but may become positive at some later time point. There are unique features of perinatal infection with HIV with respect to cure that make these infants very interesting as potential subjects for study. And the first is that the timing of infection is relatively well defined, particularly when infection occurs peripartum. In addition, in early infancy, there are very low levels of CCR5 expression on CD4 lymphocytes, which may limit the ability of HIV to spread throughout the T cell population and be an intrinsic limitation on the development of the viral reservoir. There's the opportunity for immediate intervention with suppressive ART after birth, which may additionally limit the reservoir size. And then the developing immune system may offer unique opportunities for boosting HIV-specific immunity through vaccination and other means. And we all know the story of the Mississippi baby whose mother had not received antenatal care and was not on antiretroviral therapy. And so this baby became infected, but was diagnosed at birth and immediately placed on suppressive antiretroviral therapy. You can see in panel A here how a virus load declined rapidly and then became undetectable as the baby remained on a suppressive regimen. But then the infant was lost to follow-up. And when returning to care several months or years later, it was found to be not only undetectable, but not to have any detectable HIV despite exhaustive search and testing, despite having been off antiretroviral therapy for some time. Unfortunately, after several additional months of follow-up, this baby did eventually rebound, very much like the Boston patients and some other, the San Francisco patient who Tim Henrich treated, eventually rebounded despite having undetectable reservoirs by conventional assays. And notably, at the time of rebound, the baby then seroconverted, since antibody development had been prevented by rapid suppression of virus replication at the time of birth. These are studies, of course, done by Deb Prasad and Kathy Luzuriaga. In addition, we have the example of the baby from the SHARE study who had early antiretroviral therapy, and then when treatment was eventually stopped, has now gone for many years without showing evidence of HIV infection. And as Deb Prasad has shown, the size of the reservoir in children is dependent on when antiretroviral therapy was started. These are actually adolescents who are now many years into infection and have been on consistent suppressive antiretroviral therapy. And you can see that those who started treatment at less than 1 year of age had substantially smaller reservoirs than those who started in early childhood or later in childhood. Now, there are a number of important challenges in conducting cure research in infants. First and foremost, the safety and pharmacokinetics of many of the candidate interventions in infants, children, and adolescents is unknown. In addition, particularly in infants and toddlers, the limitations on blood volume sampling make sampling for reservoir assays challenging. Most candidate therapeutic vaccines have not been studied in persons younger than 18 years of age, so some of the interventions we'd most like to test in very young children are challenging until safety data and immunogenicity data are available from studies in adults. And the risk benefit of most approaches using genetically modified peripheral lymphocytes or stem cells are really not suitable for study in children. So together with my colleagues, Roger Shapiro and Matthias Lichterfeld here at Harvard, we embarked on the early infant treatment study to identify infants who had been infected in utero and were positive at birth in Botswana, and then immediately initiate antiretroviral therapy with the goal of severely limiting the establishment of the viral reservoir, and then tracking these infants over time to see what happens with the reservoir. So this was a single-arm, non-randomized trial of early treatment in antepartum and peripartum infection. The target population was children who were within 7 days of life if they had antepartum infection, or within 2 months if infected peripartum. And we initially recruited from Gaborone and Francistown in Botswana, and then extended the regions from which we recruited. The original intention was to recruit up to 30 patients or participants from in the antepartum cohort, and up to 20 in a peripartum cohort, and 20 controls, that is infants or children who were already on ART that had been started within the first year of life, but beyond the ante or peripartum periods, and to treat these children then for 96 to 192 weeks. Babies were started initially on a nevirapine zidovudine 3TC regimen, and then at 2 weeks of life, they were old enough to be switched from nevirapine to boosted lopinavir. This study, of course, began before we had sufficient data on the safety and PK of integrase inhibitors for use in these very young infants. Through the extraordinary work of Roger Shapiro and his colleagues in Botswana, Ajibola, and Sikulele, more than 9,100 newborns were screened. This represented nearly 30% of all HIV-exposed infants in Botswana during the time we were enrolling this trial. The overall rate of confirmed HIV infection was only 0.4%, which speaks to the extraordinary success of the prevention of mother-to-child transmission efforts in Botswana. This led us to identify 40 children who had antepartum infection, as well as 23 controls. And by the time of the analysis that I'll present to you, 30 of the 40 antepartum infected children had reached 84 weeks of follow-up. As you can see in this slide, not surprisingly, the initiation of antiretroviral therapy led to the very rapid reduction in plasma HIV RNA levels, with the majority of children becoming undetectable by week 12. Of course, there were some children who had episodes of viremia subsequently because of adherence, particularly due to the challenges of tolerability of the liquid ritonavir and the lopinavir regimen. If we looked at cell-associated HIV DNA in these participants and compare them with other infants, as well as with other groups of patients, you can see that over the period of 84 to 96 weeks, there was a decline in cell-associated HIV DNA. This is by the standard PCR assay, quantitative PCR assay, not by the newer IPDA or intact proviral assay. And you can see that these levels were significantly lower at weeks 84 or 96 compared to the controlled children who were tested at around week 93. In addition, these early treated infants had much lower levels of proviral DNA compared to adults who were treated with antiretroviral therapy during chronic infection and adults who initiated antiretroviral therapy during acute HIV infection. Matthias and his group, particularly Pilar Garcia Broncano, the first author of our paper from last year in Science Translational Medicine, characterized the full length of these genomes to determine which were intact and which were defective. And you can see here a variety of these children The APs are antepartum and the PP is a peripartum infected child. And you can see the pink lines represent intact genomes. The green ones are hypermutated, and these that are a little hard to see here show deletions, reflecting that many of the viruses in the antepartum children were intact, but others were defective, as has been seen in adults. And then looking at the control children, we see many fewer intact sequences and a larger number of defective sequences. Now, if we chart the decline of intact and defective proviruses over time, you can see that along with the decline in overall the cell-associated DNA, as I showed in the previous slide, we also saw a decline in the number of intact proviral genomes and a decline in the number of defective proviral genomes in the early infant treatment cohort, so that at weeks 84 to 96, both intact and defective proviruses were at a lower level compared to the control infants. And here the open circles represent numbers below the limit of detection, and the filled circles are actual detected numbers, and the squares represent the controls, and the circles are the early treatment cohort. Likewise, if we compare them to adults on suppressive antiretroviral therapy, you can see that the number of intact viral genomes in the early infant treatment cohort is substantially and statistically significantly lower than in the heart-treated group. We looked at the rates of decay of the defective and intact proviruses and found that cells carrying intact proviruses appeared to decay much more rapidly than cells having defective proviruses. And that was highly statistically significant. Another intriguing finding was the relationship between plasma viremia and positivity in an EIA assay at week 84. Recall with the Mississippi child that child had not made its own antibody until having viral relapse many years later, because early treatment prevented the establishment of the baby's own humoral immune response. And so what's shown here in this slide, the green boxes represent plasma HIV RNA levels below the limit of detection, or less than 40 copies. And then here in the right-hand column, again, the green boxes represent seronegative testing at week 84, and the orange boxes are positive EIA tests. And what you can see is if a baby was able to achieve complete viral suppression by about week 24 or 36 and stay below 200 copies, those babies never seroconverted, even if they had low levels of detectable viremia. So for example, this baby Q over here has 300 copies at week 24, but then 166 at week 36 and becomes undetectable, remains seronegative. Whereas baby U, who has a brief spike up to 4,000 copies, seroconverts. Here again, baby W has 76 copies at week 24, but is otherwise undetectable and stays antibody negative, whereas this baby AA had become undetectable, but then has a period of non-adherence and spikes up to 90,000 and has a borderline positive ELISA. So this tells us something very interesting about just how much virus does it take in order to stimulate an antibody response, and at what time that virus has to be present in order to elicit a humoral immune response. So in conclusion, early initiation of antiretroviral therapy in HIV-infected neonates significantly limited the establishment of a reservoir in peripheral blood mononuclear cells harboring intact proviruses. Cells harboring intact proviruses decayed more rapidly than cells harboring defective proviruses. Maintaining a virus load below 200 copies prevented the development of humoral immune response to the virus. These data provide strong empiric evidence supporting the immediate initiation of antiretroviral therapy for HIV-infected neonates. Well, what are next steps? The big question is, what additional interventions are available and appropriate for use in these infants and young children? Well, we can't really use latency reactivating agents. Those are mostly drugs being developed as anti-neoplastic and far too toxic to consider use in very young children. I've already explained why therapeutic vaccines are challenging to translate into use in young infants and children. Likewise, checkpoint inhibitors and cell-based therapies are not available for use in this population. And so that leaves us really with broadly neutralizing antibodies. And so the early infant treatment study is now being followed up with the Tatello study, which is asking whether a dual combination of broadly neutralizing antibodies can sustain viral suppression in HIV-infected infants who initiated treatment in the first days of life. That study is now ongoing, and the children are beginning to receive their antibody infusions, and we hope to have results within the next year. So let me stop by acknowledging, first of all, my co-investigators, Matthias Lichtenfeld, and particularly Roger Shapiro, and the entire group of the Botswana-Harvard AIDS Partnership, who worked so hard to recruit and to care for these infants. Pilar Garcia Brancano in Matthias Lichtenfeld's lab at the Reagan Institute, who did most of the molecular assays, and the rest of our group and collaborators. Thank you very much for your attention.